Okay, good morning. Good morning to the today's chief guest, Sri Nachima Kumar, as well as welcoming for this guest lecture. Whereas good morning to the students. Today I'm welcoming to the guest lecture on the general scientific methods for designing in vivo preclinical experiments. So I'm requesting to Mr. Srinivas to give a brief profile of Mr. Narasimha Kumar. Yes, Srinivas.
Yes, sir. Thank you, sir. Myself, Mr. Srinu Talla, Assistant Professor from Department of Pharmacology, CHALAPATHI Institute of Pharmaceutical Sciences. So it's my immense pleasure to introduce our today's guest lecture speaker, Mr. G.V. Narasimha Kumar. He started his career as an assistant professor in Department of Pharmacology in Sri Padmavati School of Pharmacy in the year 2013 to 2016. After that, he joined as a research assistant in Department of Pharmacology, Siddha Central Research Institute, Central Research Institute under Siddha, Ministry of Ayush, Government of India from September 2016 to November 2018. Currently, he is working as research officer, scientist grade 1 in Department of Pharmacology, Dr. Anjali Chatterjee, Regional Research Institute for Homeopathy under CCRI, so Ministry of Ayush, Government of India. He studied his B.Pharm in 2005 to 9 in Sri Padmavati School of Pharmacy, Tirupati. He completed his Masters in Pharmacology, Sri Padmavati School of Pharmacy in the year 2011. Currently, he is doing his Doctor of Philosophy, Department of Pharmacology in SRM, Chennai. So he is very good at handling the laboratory animals and very effective in designing and carry out the preclinical toxicity studies, especially in the departments of neurology, neuropharmacology, and toxicology studies. He is the best outgoing student for the year 2011 from the Sri Padmavati Group of Educational Institutions. He awarded Rashtrapati Scout by Dr. K.R. Narayanan, President of India in the year 2002. He is qualified GATE 2009 with All India rank 952 with 96.12 percentile. He qualified GPAT 2014 All India rank with 1108. He qualified again in GPAT 2016 with All India rank 229. So he has guided 3 project students at the post-graduation level and 2 projects at the undergraduation levels. He has attended many workshops. He conducted many conferences. He has done 24 national level publications with Science Cited Index 5, Scopus Index 10. He has reported 2 case reports in his publications. He has written a handbook in microbiological assays. So he has presented at various conference levels at 6 o'clock. Today, he is going to discuss about the general scientific methods for designing the in vivo preclinical experiments. It's my pleasure to introduce the speaker.
Yes, so I'm requesting Narasimha Kumar to start our lecture.
Sir, Narasimha Kumar, sir, please unmute your mic, sir. Please unmute your mic, sir. Thank you, sir. You can proceed.
So I would like to express my thanks to Principal of Sri Chalapathi Institute of Pharmaceutical Sciences, Dr. Ramarao Nadanla sir, and HODI Department of Pharmacology, Dr. Narendra Babu sir, for giving me this opportunity to discuss about an important topic related to preclinical pharmacologists, that is general scientific methods for designing in vivo preclinical experiments. This topic would be useful mainly for these budding pharmacologists who are getting passed out, that is M.Pharm pharmacology people, for designing their preclinical experiments, mostly toxicity studies, efficacy studies, or other not only in vivo, in vitro preclinical experiments also. So why we need to design, why we need to pre-plan our in vivo preclinical experimentation. Mainly coming on to preclinical studies using animals are mainly used to study the potential of therapeutic drugs before translation of these drugs into clinical trials. And these animal studies nowadays are playing a major pivotal role in the development of science. So nowadays, what are the mainly lacunas in preclinical in vivo experiments? Preclinical experiments mainly lack of reproducibility, either in results, lack of scientific rigor and transparency in the experimental design, and lack of translatability, means the experiment carried out preclinical level cannot be translated into clinical, means the molecules or therapeutics from bench side to bedside. Translation is very, very few. Why these lacunas are there in preclinical experiments? Mostly due to experimental bias. So there are few studies which have been carried out by many of the researchers to know about what are the lacunas, mainly why the preclinical studies are lacking. So a online webinar survey conducted by Nature shows that more than 50% of the preclinical researchers have agreed that they have failed to reproduce 70% of their own experiments, and more than half of them have said that they have failed to reproduce their own experiments. So another study by Swiss survey highlights that mainly poor experimental design and statistical analysis have led to the decrease in the reproducibility, replicability of experimental studies, mainly preclinical experimental studies. So why this? What happens when there are these imprecise and biased results? These imprecise and biased results from animal experiments result in the movement of or transfer of biologically inert or even harmful substances into clinical trials, which may expose the patients to unnecessary risk. So along with this, there are wastage of resources and funds. So what are the remedy for this lacunae in preclinical experiments? Planning experimental research and procedures on animals before carrying out the study. Before carrying out the study, we need to design or pre-plan our experimental research and procedure on animals, which will ensure precision, internal validity of the experiment, and also likelihood of success of the research or experiment carried out. So for this, we need to prepare experimental protocols before the initiation of the studies. These experimental protocols are central for reproducibility, as they include all the necessary information for obtaining consistent results. Experimental protocols should be prepared by adhering to the scientific methods and all the necessary guidelines. So today we will discuss about mainly important scientific methods which are mostly responsible or should be designed before carrying out an in vivo preclinical experiments. So hypothesis and replicability. Animal models, which we will discuss, are mainly responsible for the replicability or reproducibility of an experimental design. groups, variables, sample size, randomization, blinding, and statistical analysis are responsible for the internal validity of an experiment. Hypothesis. So, a hypothesis, after initiating a research, after an extensive literature survey, we need to design our, or we need to formulate our research question, and research question will lead to the generation of hypothesis. And hypothesis is a predictive statement which can be tested by scientific methods within a specified time limit that relates an independent variable with an dependent variable. Most of the experimental studies are being carried out only to test the hypothesis. Hypothesis is the key driving point for an experimental research. For example, consumption of sugary drinks every day leads to obesity. This is a hypothesis. This hypothesis can be tested by scientific methods within a specified time limit. And within this hypothesis, sugary drinks are independent variables, and parameters related to obesity like waist circumference or lipid levels can be dependent variables. So during the generation of this hypothesis, which is the key for an experimental research, we need to answer our own selves many questions like what, who, where, when, how, why, and this leads to the development of an hypothesis. And mainly hypotheses are of mainly two categories: exploratory hypothesis and confirmatory hypothesis. Exploratory hypothesis, the name itself indicates exploratory hypothesis leads to generation of an hypothesis, which are intuition driven. And exploratory hypothesis is a lifeblood of preclinical science. In exploratory hypothesis, the experimental design cannot be rigid. Experimental design can be made flexible. And for example, in case of exploratory hypothesis, like means if we are going to find out a pathophysiology of a disease or how a disease occurs, or the conditions which leads to the production or generation of a disease, these all comes under exploratory hypothesis. And in case of confirmatory hypothesis, the confirmatory testing, the hypothesis generated in exploratory hypothesis will be subjected to testing. That is nothing but confirmatory or hypothesis testing, where we need to follow rigorous experimental design, which will give us the proof of concept related and proof of preclinical efficacy. For examples of exploratory hypothesis, defects in insulin signaling pathways leads to development of type 2 diabetes. This we have found out by doing an exploratory hypothesis in any animals or somewhere. And in order to validate this exploratory hypothesis, we will go for hypothesis testing. That is nothing but confirmatory hypothesis. Example, drug A reduces blood glucose by increasing insulin receptor sensitivity. And this is our confirmatory hypothesis, which we will verify in an animal model by applying rigorous scientific methods. And confirmatory hypothesis, while doing our experiment, we need to postulate in confirmatory hypothesis two types of hypothesis: null hypothesis, which is normally indicated by H0, alternative hypothesis, which is normally indicated by HA. For example, in this example, we can see that application of a biofertilizer X causes increase in plant growth, which is our alternative hypothesis. And null hypothesis, application of biofertilizer X do not increase plant growth. Normally, we propose both null hypothesis and alternative hypothesis, and the researcher tries to unprove null hypothesis. Means the researcher tries to disprove or nullify null hypothesis, so that automatically alternative hypothesis will be proved. For example, in the previous example, drug A reduces blood glucose by increasing insulin receptor sensitivity will be our alternative hypothesis. Drug A do not reduces blood glucose by increasing insulin receptor sensitivity will be alternative hypothesis. And in our animal model or in our experimental design, we will try to disprove disprove null hypothesis, such that automatically alternative hypothesis will get proved. Means we will try to show that by giving the drug A, there will be an increase in the insulin receptor sensitivity, which we can show by measuring the levels of blood glucose or measuring the levels of insulin or insulin receptor expression and etc. So next, coming on to animal model. So in animal models also, mostly we will not be using the wild ones. We will be going for lab animals. Why laboratory animals should be used in experimental research compared to wild ones, which have more variability in their in their genetic structure and also phenotypic structure. Lab animals are more homogeneous, means they won't have much of the heterozygosity, and their species identity is known, and their uniformity in gene chemistry and their mating characteristics, means the parental history will be known in case of laboratory animals, and we will be giving a designed and balanced feed in case of laboratory animals, and hygiene will be ensured. Almost they will be free from disease. We can obtain specific pathogen free animals, and mostly the laboratory animals are docile, easy to handle. Means I would like to say that the experimental conditions while carrying out research in case of laboratory animals can be maintained, while in case of wild ones, it is very hard to maintain the experimental conditions. Animal model. Choosing a suitable animal model for our experimental design is the heart for our research in case of preclinical research. Most of the uh, researchers are mostly when come to colleges, we will be choosing the animals which are easily available, which are easy to maintain. Mostly we will be doing our studies on rats because which are easy to handle, easy to reproduce. But it is not so. We have to choose an animal model depending upon the research question and our hypothesis. So when coming to animal model, first of all, which strain and a substrain of animals we need to use. The choice of this substrain or strain will be based upon our research question, whether we are going to carry out toxicity studies or efficacy studies, and whether we require heterozygosity or homozygosity, means variability in our research results or uniformity in our research results. Mainly, there are two types of strains. Animals can be categorized into two types of strains: outbred strains, inbred strains. Outbred strains, with the name itself indicates, their parents are not closely related and they are heterozygous both genetically, and their response to treatments are also will be different. Means, for example, outbred strains rats, Wistar rats are Sprague-Dawley rats. Though if we obtain these rats, Wistar rat or Sprague-Dawley rats, from two different vendors, we cannot say that they are same. Though their name is same, but they're genetically and phenotypically they may be different because they are outbred, because they are not produced from parents who are closely related to each other, and the response to treatment in them will be different or variable. Whereas in case of inbred strains, they have less variability because inbred strains are produced mainly from sibling mating, means the parents who are closely related, brother sister mating. And in case of inbred strains, they are genetically and phenotypically less variable and mostly similar. Mostly used inbred strains are mice. And sex. So after selecting a strain or substrain of the animal based upon our research question, next comes the question is sex. So which sex of animal should be used in our experimental design? So most of the researchers have a misconception that females are intrinsically more variable, so our results may be variable. So in order to produce a reproducible results, they will go for mostly using males in their experimental design. This is a misconception that female, mostly either mice or rats, females are more intrinsically more variable. But a recent meta-analysis shows that variance seen with males and, for example, variability is more in case of males compared to females. Here we need to understand that when variance is more, we cannot find reproducible results. If reproducible results cannot be produced, we cannot say that experimental research have produced a good chance of success. Means the experiment which we have done with an animal in our lab, if it cannot, the same results cannot be produced in another lab, then we can say that the results are variable, and we can say that the results are not reproducible. So in order to it has been hypothesized that these results will be variable more in case of female mice, and hence mostly male mice were used in most of the experiments to produce reproducible results. But this selection has a bias, because when we use a specific type of sex, for example, male or female, we cannot successfully translate our research findings to both sexes. So we need to take into consideration both sexes into our experimental design, unless the research question or hypothesis demands a specific sex. For example, in case of male reproductive toxicity studies, we will not go into using female animals, and when we are going to evaluate any medication in the treatment of diseases related to females, we cannot use males. So except to these exceptions, it would be better if we are going to use both sexes in our experimental design, which will lead to successful translation of our research findings into clinical level also. Next, coming on to the age of the animal. Which age of the animal should be selected in our experimental design? Age of the animal in turn depends upon the biology of the disease which we are going to study. For example, we are going to study regarding Alzheimer's disease. It won't be apt to select early juvenile animals. It would be apt to study in old animals, means the aged animals, after aged animals having age of minimum 1 year and or etc. Because different organs and systems will reach developmental maturity at different times, and precise age is most relevant for the research question to be considered carefully. For example, if we want to study about juvenile diabetes, it won't be feasible to study in adult animals. We have to take the suckling animals, and we have to study the juvenile diabetes in suckling animals. So age plays a major role. And when coming to mostly the animals which are extensively used in our preclinical research. In preclinical research, mostly we know that rodents are mostly used. In rodents, mostly the mice are the animals which are mostly used in preclinical experimentation. The slide which we are seeing is the comparison of the number of animals which have been used over the few past years. In case of mice, if we can observe that compared to 2000 year in 2018, there has been threefold increase in the usage of mice. In case of mice, mostly inbred strains compared to transgenic, knockout and congenic, because inbred strains, as we have discussed, they will give us reproducible results, which because of their inbred in nature and due to their less variability. In case of inbred strains, which mice are mostly used, C50B, C57BL/6 are the animals which are mostly mice which are mostly used, followed by BALP/C. Why these animals are mostly used? Due to their inexpensive care, high genetic similarity to humans, quick reproduction, and mice are the first animals to be fully genome sequenced after humans. In mice, C57 BL/6 mice was sequenced, their genome was sequenced in the year 2003, immediately after the humans. So in case of animal models, in case of mice, most commonly used models are animals are C57BL/6 and BALB/C mice, which we can see in most of the published research papers. Why C57BL/6 or BALB/C? These are most easily available, and mostly they have a common factor. They are inbred, means less variability, and they can offer reproducible results. And C57BL/6, as we have discussed, it was the first mouse whose genome was fully sequenced after humans. And C57BL/6 mice has traceability and easy breeding techniques. They can be bred easily, and their sexual maturity acquires within a month, means after 4 weeks, and they can be bred up to 1 year. Their litter size is approximately between 6 to 10, and they can produce 4 to 6 litters in their whole lifespan. So they can be easily used. And C57BL/6 mice are mostly used in the most of the physiological or pathological models for in vivo experiments. For example, most of the researchers will be using rats in diabetic experimentation, but we know that rats has a diffuse pancreas and they are not suitable. But C57BL/6 animals are mostly used in diabetics research, and they are mostly prone to diet-induced obesity and diet-induced type 2 diabetes mellitus. So they are most apt animals to carry out this research. And coming to BALB/C animal, BALB/C is also inbred, albino, and mostly it is an immunodeficient model. So that's why BALB/C animal mice is mostly used in cancer therapy, infectious diseases research, and also immunological. If we are to if we are to evaluate any drug for immunomodulatory studies or infectious diseases or cancer therapy, it will be apt to choose BALB/C because they are more tumor prone, and even hybridoma and monoclonal antibody production is being done from BALB/C mice. Even in case of, for example, now coronavirus disease, humanized BALB/C mice have been used as an animal model to mimic coronavirus research, SARS-CoV-2 disease. If we are not able to, these humanized BALB/C mice are currently being produced in the Jackson Laboratory, America. So this BALB/C mice offers more similarity to human physiology and anatomy. Compared to mice, rat are the next to mice rodents which are mostly used in our preclinical research. Mostly we have two strains of rats, Vista rat and Sprague Dolly rat. They are mostly outbred. Outbred means they are produced from parents who are not closely related to each other. They are heterogeneous and they offer more variability. So where this star rat or Sprague Dolly rat mostly find their applications, research applications, mainly in neurobiological research and toxicity studies. If we go through OECD guidelines for toxicity studies, we can observe that rats are mostly preferred. Why rats are mostly preferred? Because they're outbred, heterogeneous. As if we can see, in case of toxicity studies, we need to obtain results from a wide variety of population. So it would be apt to test our medicine for its safety in an animals which offer more variety or variability. So in case of toxicity studies, rats are preferred. Whereas in case of in vivo efficacy studies, where we will be testing our drug on a specific disease, means we are limiting our population to a specific disease. So we require an animal model which produces the disease, and this animal model should be reproducible, means it should be genetically less variable and it should be homozygous. So in case of most of the preclinical research experiments, mice will be used mostly in the efficacy studies compared to rat, and rats are mostly used in the toxicity studies due to this nature. So while deciding our preclinical research experimentation, we need to take into consideration the research question and hypothesis to select an suitable animal model, followed by the suitable species, sex, age of the animal. Next, coming on to experimental units. Mostly in case of our experimentation, we will be housing the animals in a cage, 6 animals in a cage or 10 animals in a cage. We have to consider that experimental units are the number of animals. So in a cage, if we are giving the treatment in through water or feed, we need to consider the whole cage as an experimental unit because there is no variation. We are giving the treatment in case of a through water or feed means all the animals are equally, they may be receiving the drug or unequally. So in case of this type of studies where our treatment or intervention is through diet or water, entire cage has to be considered as an experimental unit. Whereas if we are giving each individual animal separately the intervention through injection, then each individual animal will be considered as an experimental unit. In the same way, as we can see, in case of the zebrafish which are present in the tank, they will be mostly given medication through the water dispersed in that. Then this entire zebrafish tank will be considered as a single experimental unit. Same applies to the litters which we are seeing. The entire litters will be considered as a single experimental unit. So while designing our experimental study, we need to be very clear that our experimental unit is a single animal or a single cage or a litter. We need to be clear with respect to that. Next, coming on to groups. Grouping of animals is also very fundamental in experimental design and should be preferred than historical comparison. Historical comparison means most of the people we in colleges, earlier I was also working in a college, we will reduce, in order to reduce the financial burden, we will take the data from the previous studies with respect to general control, that is normal control group or vehicle control group or disease control group, but it should not be done. While designing experimental research, all the experimental groups should be, the research should be carried out with all the experimental groups at a time. No historical comparison should be done, which will lead to experimental bias. Normally, we can see these type of groups in our experimental research. Vehicle control should be very must, and which should be, the group will be administered with vehicle. Positive control, for example, if there is a standard treatment, for example, in case of diabetes, there is a standard treatment. Then we should include a group where we are expecting the desired changes. That will be the positive control group. And negative control group. In case of some disease, there is no standard treatment available like SARS-CoV-2. Then we should include a negative control group in which we won't be expecting any desired changes. And next, coming to sham control, groups are normally which are used to mimic a procedure or treatment. Next, coming to variables. Variables are very, very important parameters which we should consider during our experimental design. Consider this example, effect of drug X on blood glucose levels in diabetic mice. Here drug X is the independent variable, means the variable which will be manipulated by the researcher, and blood glucose levels is the dependent variable which depends on the independent variable, means depends upon the drug X, the blood glucose levels will be changing in the diabetic mice. Apart from this independent variable and dependent variable, the researcher has to keep in mind the of nuisance variables. Nuisance variables are very, very important and which can influence the dependent variable. For example, in case of this experiment, nuisance variable may be feed, maybe water, means in this diabetic mice, we may be giving the feed which is less less sugar. It means which is which the feed which contains less sugar content, less lipid content, which may affect our the blood glucose levels. So due to these nuisance variables, if present in a experimental design, we cannot clearly say that the independent variable has led to the changes in the dependent variable. So we need to nullify, or we need to take care of the nuisance variables in our experimental design. Nuisance variables are also called as confounding variables also. And coming on to dependent variable is nothing but the outcome measure. So outcome measures, as in case of clinical research, we should define outcome measures both as primary outcome measure and secondary outcome measure. Primary outcome measure is the one which will decide the result of our research, which is very, very essential to conclude experiment. Secondary outcome measures are ancillary along the measurements which can be made along with primary outcome measures, but which should not be used to conclude an experiment. For example, in the previous example, effect of drug X on the blood glucose levels in diabetic mice. Blood glucose levels are our primary outcome measure, which will define the conclusion of our experiment. Whereas secondary outcome measures may be measurement of insulin levels, lipid levels, or liver profile, which may give an additional information about the effect of this drug X in diabetic mice, but which should not be used to conclude the experiment. And in case of dependent variables, there are mainly two categories: continuous variables and categorical variables. Continuous variables are quantitative, are expressed in numerical scale. Categorical variables are qualitative. Why we are discussing about variables? Because these variables in turn are very much essential in choosing the appropriate statistical analysis for our experimental design. So whether we need to choose a parametric test or non-parametric test is dependent upon the whether we in our experiment we have a continuous variables or categorical variables. Continuous variables, example, blood weight, body weight, BP, blood glucose. Whereas categorical variables, binary variables, where the outcome will be in two conditions, whether yes or no, present or absent. Ordinal variable, where the response will be noted in case of like mild, moderate, severe. And nominal variables, where the response, behavioral response, means the time spent by the animal in dark side of the cage or light side of the cage. These will come under nominal variables. Coming on to nuisance variables. Nuisance variables are mostly neglected in our experimental design. Means no one will be taking care of these nuisance variables. But as we have discussed earlier, these nuisance variables will influence the result, our result, and it will influence the dependent variable, outcome measures. So nuisance variables are the other sources of variability which may influence the outcome measure. So nuisance variable leads to increase in the variability of responses. And while designing our experimental design, we need to take care that these nuisance variables are blocked in our experimental design. We need to take care that the experimental design, the nuisance variable should be blocked. Examples of some we will discuss with an example. For example, we know that age can affect behavior. So we are designing an experiment looking at the effect of dopamine agonist on behavior. So we know that behavior will depend upon age. If we select the animals, for example, rats, having a wide range, then age is a nuisance variable, because we know that adult animals are more aggressive and they are not docile, whereas aged animals will be somewhat docile and calm. So while designing our experiment, if we are not taking this age, the nuisance variable in this experiment, and if we are not blocking that, then it will lead to variability in results. Means in this experiment, we have to choose a specific age group of animals. That is, whether the aged animals or adult animals, we have to choose. And even we cannot imagine that location of a cage of animals within the room is also a nuisance variable. For example, if you are placing the cage of the animal near to the door, frequently the door will be opened, the animals will be subjected to a lot of stress due to opening of the door, which may be due to sound or something. So the animals which have been placed near to the entrance of the room will be subjected to unnecessary stress, which is a nuisance variable. Weight of the animals. In most of the experiments, the without considering the weight of the animals, low weight of animals, for example, 90 grams of rat will be taken into the experiment, and also 200 gram of rat will be taken into the experiment. But this weight of the animal is an variability, is a nuisance variable, which we need to account for. In the same way, time or day of the experiment, even the investigator, these all nuisance variables will lead to bias in our experimental design. And while designing it experimental design, we need to know about what are the nuisance variables which will be affecting our experiment, and we need to take care of all these nuisance variables such that they are blocked from the experiment, so that we can say that our experiment has an internal validity. Next, the most important part of our preclinical research experiment is the sample size, means the number of experimental units per group. Mostly, many researchers consider 6 animals per group as an adequate sample size. But considering the review of literature and available literature on this issue, 6 animals per group has little scientific and statistical basis. Means it is not suggested that every experiment, every in vivo efficacy experiment should be conducted with 6 animals per group. The number of animals per group, that is sample size, is dependent upon the research question, the hypothesis, animal model selected, number of groups, and also it also takes into the account the nuisance variables also. So most of the studies which have been published, they don't contain any sample size justification. And if we are not publishing, and due to this, the proportion of published papers, they are publishing false reports. Means if we are concluding that the drug has effect on X disease with using 6 animals per group without taking into consideration the groups, animal model, the confounding variables, and statistical analysis, it will lead to nothing but a false positive report. So in case of sample size, if we take very few animals in our group, it will be a total waste. The experiment is of waste and it will not lead to any conclusive result. Whereas if we use too many animals, it will be a partial waste and unethical killing of our usage of animals. So methods for determining sample size. Why I am stressing on sample size means when we are going to publish our results in an peer-reviewed journals, for example in Web of Science or Scopus, nowadays as per ARRIVE guidelines, animal research reporting of in vivo experiments, justification for sample size should be reported in the paper. Means we have to justify why this amount of samples, means why this number of animals in the groups have been chosen. So what are the different methods for determining sample size? First method, traditional or common sense method, most of the researchers follow in college settings, even in the research labs, means they will choose the sample sizes based upon the previous published papers or reports. Means if I am going to work on a model of streptozotocin induced diabetes, then I will go on to the review of literature related to streptozotocin induced diabetes in mice, and I will use the animals which have been used in the previous papers. If they have quoted 6 animals or 7 animals per group, citing that research paper, the researchers will use the same number of animals. But this traditional common sense method has an experimental bias. We need to consider that the experiment which has been conducted by that specific researcher is specific to that lab, that experimental conditions, and specific to that investigator. Whereas now we are conducting an entirely different experiment. We need to take into account our research question, our experimental conditions, our biases, and etc. and therefore we need to standardize the model in our experimental conditions. Then we have to choose the sample size. So traditional or common sense method is mostly used, but it is not scientifically justified. Second method is resource equation method, but this method is mostly used in agricultural and industrial research using an equation which is not of our interest. But the most important method by which sample size is determined in conventional research is by power analysis. Power analysis is the most important technique by which scientifically sample size can be determined in a preclinical research design. Not only in preclinical research, clinical research, it is a very famous term, power analysis. It was introduced by Jacob Cohen in the 1960s. It is a mathematical relationship between 6 variables. So before going on to how we can determine a sample size, we need to discuss about few terms which are of very important. First thing, effect size. What do we mean by effect size? The name itself indicates effect. For example, there are two groups, a normal control group and diabetic control group, and we have given our intervention in these two groups, and the difference in the blood glucose levels between these two groups is nothing but the effect size. For example, in normal control group, the blood glucose level is 120 mg per deciliter, and in the diabetic control, it is 150 mg per deciliter. Then 30 is this effect size. Means effect size is the difference between means of the two groups which are being compared, which would be of interest biologically and which would be worth taking forward work for clinical trials. So the idea about this clinically significant difference between the groups should be taken preferably from previously published studies. So effect size normally is indicated by M1 minus M2. M1 is nothing but means of different groups, two different groups, which are of our biological interest. The effect size is the minimum effect that is considered to be important. For example, we are conducting an hyperlipidemia. We are evaluating the effect of a drug on triglyceride levels in an hyperlipidemic animals. So what will be the minimum changes in the lipid levels, for example, triglyceride levels, which we see as an biologically considerable change? Means compared to hyperlipidemic group, the treatment given group, if the triglyceride levels are 30 mg per deciliter less than the hyperlipidemic drug, means a change of 30 mg per deciliter, if our intervention is able to bring about a change of 30 mg per deciliter in lipid level, in triglyceride level, then it is of biologically interest, and it will lead to, we can say that there has been a effect of intervention on blood triglyceride levels. Then this 30 will be chosen as an effect size. So effect size is the minimum effect that is considered to be important. Effect size is very important for doing sample size calculations using power analysis. This effect size, we can this idea can be, even we can get this idea, that is clinically significant difference between the two groups, from the previously published studies also. For example, in an experiment looking at the effect of a drug on blood pressure, the outcome measure would be systolic blood pressure, and the effect of interest could be change in systolic blood pressure. Here we consider that the effect size of 20 mmHg implies that researchers are interested in any change in systolic blood pressure greater than 20 mmHg, and an effect size smaller than this would not be worth taking forward for further research. So this effect size, normally, if we are not having an data of effect size, we can go for standardized effect size, that is Cohen's d, in case of power analysis. Cohen's d is a standardized effect size. It represents the difference between treatment and control means calibrated in units of variability. Cohen's d is calculated as the difference between the two means divided by standard deviation. For example, from previous study, we have found out that when we consider this example, related to this example, systolic blood pressure was from previous studies was found out to be 130 plus or minus 30. So biologically relevant effect size was found out to be 20, and Cohen's d can be calculated as 30 divided by 20, 1.5. So this 1.5 will be the Cohen's d, that is standardized effect size. So if we don't have any, consider that we don't have, it is a very new experiment, we don't have any previous published papers from where we can calculate the standard effect size, then in this condition we can use standardized Cohen d values which are normally used in sample size calculation. If no information is available to estimate the variability or standardized effect size, a standardized Cohen's d values may be used instead of a biologically relevant one. So Cohen's d, the original guidelines suggested by Cohen in the field of social sciences, suggest that effect size as follows. For example, Cohen considered small effect size 0.2 as small effect size, 0.5 as medium effect size, and 0.8 as large effect size. However, in work with our laboratory animals, the values shown here 0.2, 0.5, 0.8 are not accepted, and they can be marginally moved to the higher size. So in case of preclinical experiments, Cohen's d values have been put upon higher side. That is, Cohen's d, if you say that 0.5 is the effect size, 0.5 as small, 1 as medium, and 1.5 as large. Here we need to understand that the smaller the effect size which we need to observe, which we are aiming to observe, the larger will be the sample size. Means if we are finding to observe a small difference between two groups, we need to take a large number of animals to as sample size to detect that small change. For example, we wish to detect a small change of 2 mm of Hg difference in systolic blood pressure between two groups, that is normal group and treatment group. Then to observe this small effect size of 2 mm Hg, we need to take a large number of animals as sample size. Whereas if we wish to observe a large effect, means even if we consider that a difference of 30 mm Hg or 40 mm Hg is biologically relevant, then it will be, we require a very small number of sample size animals, means very less number of animals in per group to detect that biologically relevant effect of 30 mm or 40 mm Hg. That's why we can see that in case of clinical trials, we can see that the number of sample size per group in case of clinical trials will be very, very large. For example, 100 number of patients per group or 200 or 300. Whereas in case of preclinical experiments, it will be very, very less. That is 6, 10, 12. Why? Because this is mainly due to effect size, Cohen's d. And next, coming on to standard deviation. Standard deviation measures the variability within the sample. Information about standard deviation of a particular variable can be taken from a previously published studies, as we have in the previous examples. Systolic blood pressure 130 plus or minus 30, that 30 is the standard deviation. Power. Now coming on to the important aspect, power. Power is the probability that the experiment will correctly lead to the rejection of a false null hypothesis. Means the power ensures that the experiment which we are doing is correct. Means the result which we have got is correct can be ensured by this power. Usually in case of preclinical experiment, a power of 80% to 90% is usually specified. But we can see that most of the published papers in currently, they are underpowered. Underpowered means those results are false positive. we need to take care that underpower experiments will lead to false positive results or reports. Significance levels or p-value, mostly we are familiar with are type 1 error or alpha error. This is the probability that the experiment will produce a false positive result. So it is usually set at p less than 0.5, means that there is a 5% of chance of making a type 1 error, means there is a 5% of chance of we are rejecting the hypothesis when it is actually true. Next, the sideness of the test. So we might have heard about one-tailed test and two-tailed test. What does this mean? A two-sided test means, for example, if we are giving a intervention to a diabetic animal, if two-sided test means we can imagine that our intervention can lead to decrease in the blood glucose level or increase in the blood glucose level. So if we are not sure that whether our intervention will lead to decrease in the blood glucose level or increase in the blood glucose level, then we will go for two-tailed test. But if we know that our intervention will definitely lead to increase in the blood glucose level, then we will go for an one-sided test or one-tailed test. Mostly we have mostly softwares which can do this power analysis. Most usually frequency softwares are G-Power, R-Project, power and sample size software. Now, with an example, we will do this sample size calculation using power analysis. So this is a bit complicated issue, the sample size analysis using power analysis, but you have to go extensively through the review of literature and you have to understand that is very most important to do sample size calculation using power analysis so that our studies, the internal validity of the experimental design will be maintained. Consider this example. Effect of drug A on triglyceride levels in hyperlipidemic mice. Here I have considered effect size of 0.5. 0.5 effect size means earlier we have seen that 0.5 effect size is a small effect size as per Cohen's d. And here I have set the significance level at 0.5 and power I have set at 85% power. And two-tailed test means I don't know whether my drug A can increase triglyceride levels, and number of groups, only 2 groups, normal and hyperlipidemic mice. Statistical test, as we have only 2 number of groups in here, I will go for an student's t-test. So using all these parameters, we will go for sample size analysis or power analysis using G-Power software. This G-Power software is very free and we can download it from our internet. Here you can see that I have given all the inputs: test family t-test, statistical test, means difference between two independent means, type of power analysis. This is a priori analysis, means we are computing sample size even before the experiments, and we are determining the sample size by inputting all these parameters: tail, two-sided tail, effect size D 1.5, alpha error 0.05, power, probability of error 1 minus beta is 0.85. And here when you have calculated, here you can see that the sample size, group 1 10 animals, group 2 10 animals, total sample size is 20, and actual power which we have got is 88.6 compared to the power which we have computed 85.
మళ్ళీ నెట్ కనెక్ట్ అవ్వాలేమో సార్ ఒకసారి చూడండి.
And if we change the power, one-tailed test, previously we have seen regarding two-tailed test. If we come to one-tailed test, if we in the same experiment, if we go for a one-tailed test, and that means we are sure that whether our drug may increase lipid levels or decrease in lipid levels, then we will go for one-tailed test. Then there is a decrease in the number of animals. So the number of animals. required in a group are 8 and 8, and total number of animals required are 16. So this power analysis helps us in determining the sample size, and we can justify that the sample size taken in our experimental design is as per scientific methods, considering the statistical analysis also. Finally, after computing the size of animals, sample size with power analysis, we have to take into consideration the attrition or death of the animals also during the experiment. So final sample size should be adjusted for expected attrition or death of the animal. So correct sample size, there is a formula. Corrected sample size is equal to sample size divided by 1 minus percentage attrition by 100. For example, suppose sample size calculated by software is 10 animals per group, and researcher is expecting 10% attrition, means he is expecting that 1 animal out of 10 animal may have the chance of death during the experiment. Then final size sample will be 11 animals per group based upon the above formula. So when we have carried out the power analysis and we have defined our sample size, we need to include a statement in our during the publication of the report or publication of the thesis. This statement will be as such: power analysis followed by expected attrition shows that sample size of 11 mice per group has 88% power to detect an standardized estimation size of 1.5, assuming a 5% significant level and a two-sided test. This statement is now mandated by all the publishing journals of high indexed value and which are following the peer review guidelines, mostly as per the prepare and arrive guidelines. So while publishing the reports of our experiment, now it is very much mandatory to give justification for sample size, and this justification and designing of the sample size can be easily acquired by using power analysis, though it requires a little bit study on that. Next important part of our experimental design is randomization. Randomization assures that there is no selection bias among the allocation of animals to the treatment groups. So randomization of a process is used to ensure that each experimental unit has an equal probability of receiving a particular treatment. So main purpose of randomization is to prevent bias, whether it may be intentional or unintentional. So randomly assigning the experiments to groups will decrease this bias and will ensure that inherent and inescapable differences between experimental units are spread among all the treatment groups with equal probability. So randomization is also very simple to do. In case of complete randomization, we can do it with Excel, Microsoft Excel, using computer rand function. Complete randomization will be done when the animals selected are of same strain, same sex, same age group, then we can go for complete randomization. Whereas if we have some nuisance variables such that we have selected different age group animals, different sex, means both male and female, then we have to go for block or stratified randomization. Block or stratified randomization ensures that the nuisance variables or confounded variables are blocked, means if we are having both males and females in our study, we have to separate males and females, and while allocating to the groups, from males has to be randomized and female has to be randomized separately, and these randomized males and females has to be allocated to different groups. Next most important thing is blinding. Blinding is a research strategy that involves keeping the people who conduct the experiment, assess the outcome and collect the data unaware of the treatment allocation. This will lead to elimination of subjective bias in a treatment. For example, while conducting an behavioral analysis experiments, for example, assessing the behavior of an animal after giving an intervention, if the researcher notes that the group in which he is administering the intervention and the group which they are not administering the intervention, there will be a predefined subjective bias within the researcher. There may be chances of recognition of results leading to false positive reports. So in order to remove this element of subjective bias, we need to carry out blinding. Means during the allocation of the animals to the treatment groups, the blinding should be carried out. Means the experimenters or researchers should be concealed regarding the experimental groups, and while assessing the outcome also they should not be known that which group is receiving which treatment or which intervention, and while analyzing the data also they should not be having any, they should be blinded with respect to the groups. This blinding ensures that the bias, subjective bias is eliminated, and this will lead to the internal validity of the experiment leading to reproducible results. Next most important thing is statistical analysis. What we do in most of our experiments, we will design the experiment, we will carry out the experiment, then we will go for statistical analysis. It is very, very wrong. As per the Ronald A. Fisher, whose experiments have led to the development of statistics, he says that to call in the statistician after the experiment is done maybe no more than asking him to perform a post-examination. He may be able to say what the experiment died of. Means before carrying out the experiment itself, we need to select what type of statistical analysis we need to carry out for our experimentation. This choosing of particular statistical test depends upon how many number of groups are we taking, the sample size, the randomization procedures, and what are the variables, whether we are measuring the continuous variables or categorical variables. These all our parameters will help us in choosing the exact statistical analysis. So choosing a statistical analysis strategy depends upon outcome measures, number of outcome measures, property of outcome measures, independent variable of interest, even nuisance variables. For example, if there are only two groups, then simply we need to measure the difference between the average mean of two groups. We can go for a student's t-test. For example, if we have more than two groups and we are measuring a continuous variable, then we should go for way analysis followed by post hoc testing. And if we have more than two groups and we have repeated measurements, then we can go for two-way analysis. So the statistical analysis depends upon our experimental design, and in priori fashion we need to decide what statistical analysis we need to input in our experiment. So what is the take home message of this uh, webinar? So planning of an experimental design is very much vital. This leads to an increase in the scientific validity, reproducibility, and animal welfare. And in order to stick to this experimental design, we have many guidelines. For example, prepare guidelines, which are mandated, which are designed for following the guidelines which has to be followed during the designing of an experiment. Stair guidelines, frame guidelines, etc. Thank you. So I would like, if any questions or any queries from the respondents, I will be happy to answer them.
Yeah, thank you, Mr. Nasima Kumar. Students, do you have any questions? students, you can interact with that speaker regarding any information on this preclinical experimentation.
So I would be happy to answer if there are any queries.
There is no much more questions. Then we'll conclude this session. So on behalf of Department of Pharmacology and Chalapathi Institute of Pharmaceutical Sciences, we are thankful to you, Nasim Kumar, for giving such an informative session on this preclinical experimentation. Hope we will be having the support from you endeavor also.
Thank you, sir.
Okay, sir. I am concluding this session right now.
Thank you.
Thank you.